Hi everyone, my name is Kenya and I'm a researcher at the University of Sydney. So I'm a microbiologist and I do a lot of research on the antimicrobial properties of Australian honey. And today I'm going to be telling you about a recent study that we did on native stingless bee honey. So why do we study honey as an antimicrobial in the first place? Well, many of you probably know that we're facing a really serious global health crisis called antimicrobial resistance. That is basically where bacteria and fungi that we were once able to kill with common antimicrobial drugs are developing resistance to these drugs, so they're no longer as effective. And it's estimated that by the year 2050, antimicrobial resistance could be responsible for around 2 million deaths per year. And the rate that we are developing new antimicrobial drugs is just not able to keep up with this problem. So because of this, scientists have really been turning to natural products as a potential solution, because these natural products have usually evolved their properties over millions of years and they can be quite complex. So honey is one of these natural products. It's a great candidate for medicinal use because it has a long historical use of being used for that reason. Most cultures that have ever existed have recognised the properties of honey. You know, it's in ancient Egyptian texts. It's been used by indigenous cultures in Australia. It has a very broad spectrum of activity, so it works against lots of different types of things. It is multifunctional, it's non-toxic, and it can be sustainably produced. So before we get into some of the data, just like a quick refresher on how honey's antimicrobial properties actually work. So the most important thing about honey is that it's really multifunctional. So it's got lots of different things working together at the same time. So for example, we've got high sugar, which will basically dehydrate bacteria. We've got low water and acidic pH, which makes it very hard for them to grow on. We've got all sorts of compounds that come from the bees, so things that the bees add to the honey. The main one here is hydrogen peroxide, but there's also other sorts of antimicrobial peptides. We've got things that come from the plants that the bees forage on, so we've got phenolic compounds, which is what gives the honey its dark colour. These come from plants. And in the case of Manuka honey, MgO or methyl glyoxal. And then increasingly research is showing that we also have things that come from microbes. So these are compounds that are produced by microbial fermentation, whether that's in the bee gut or whether it's sometimes in the honey itself. And what's really great about this is that it is a multi-attack strategy. So if you think about a traditional antibiotic, it generally does one thing, and that means it's reasonably easy for bacteria or fungi to be able to overcome that one thing. But when we're talking about like 10 or 20 different things happening all at the same time, it becomes virtually impossible for the microbes to be able to develop resistance to that. So, it would be like having to win the lottery 10 times in a row instead of just once. It's really, really hard for them. So a lot of research, of course, has looked at the antimicrobial properties of European honeybee honey, including by me. We know quite well its antimicrobial properties, but reasonably little has been done on Australian native stingless bees. So this isn't the first study by far, but it's the most comprehensive one to date. So we focused on 3 species. We looked at Tetragonula carbonaria, Tetragonula hawkinsi, and Ostroplegia australis. So the honey made by these bees is known as sugarbag, and it's been valued in indigenous culture for a long time, so they've used it both as a food but also as a medicine, so to treat things like sore throats or itchy skin or sores. And the honey itself is quite physically different, so it's thinner, it's runnier, it's got a tangy flavour, it's quite dark in colour. So we wondered if that meant that its antimicrobial properties would be quite different from Apis honey. So what we tested, so we took 48 samples of honey collected from colonies between 2021 and 2024. Most of these were carbonaria and hawkinsi samples. We only had a few Ostroplegia samples. And we tested them against different kinds of bacteria and fungi. So you can see some pictures of these on the slide here. We've got 2 bacteria, Staphylococcus, which causes skin infections, E. coli, which causes food poisoning and UTIs, and then 2 fungi, so Cryptococcus, which causes some really serious systemic infections, and Trichophyton, which causes things like athlete's foot, ringworm, and other tinea infections. And we measured their antimicrobial strength as MIC, so that just means the minimum inhibitory concentration. Basically, we take honey, we dilute it to different concentrations in water, and then we look at the smallest amount of honey that's able to kill the bacteria. We looked at it straight up, just in water, and then we also used heat treatment to separate out peroxide versus non-peroxide activity. So I'll go into that a little bit more. Sorry, I'm just going to put the laser pointer on. Okay, so what you can see on this slide here is some graphs of our results. So basically what we've got here is our 3 different bee species, Carbonaria, Hawkingsi, and Australis. Each of these dots is a different honey sample. Each of these columns is a different microbe. So what you're looking for here is the shorter the bar, the more active on average the honey was. So down here we're looking at 4% honey is able to kill the microbes. Up here we're looking at like 32% honey is able to kill the microbes. And what we found is that every single sample that we looked at actually had quite significant antimicrobial activity and against pretty much all of the organisms. So that's really quite unique. When you take any particular honeybee honey, it's quite variable from batch to batch, but what we saw was very consistent across all of the stingless bee samples that we looked at. It was particularly good against this last column here, so those are the fungi that cause athlete's foot and ringworm, but it was also very good against the bacteria as well. And pretty much all of the samples we looked at had, I would say, comparable or better activity than your average medicinal honeybee honey. And where it gets really interesting is after heat treatment. So in most non-manuka honeys, the main antimicrobial component is hydrogen peroxide. So the bees add an enzyme into the honey. When the honey is diluted, they start producing hydrogen peroxide. And hydrogen peroxide is very sensitive to heat. So when you heat a normal honey, it's going to destroy that enzyme. It's also going to destroy any residual peroxide activity. So that's why when you leave honey around for a while, or if you heat it up, it loses some of its properties. So what we did is we heated up this stingless bee honey to 80 degrees for 30 minutes, and then we tested it again. And what we found is that it actually retained quite a significant amount of activity. So you can see that that was the best in the Carbonaria honeys for the bacteria, that they're actually still able to kill the bacteria at relatively low concentrations. So that means that there is something unique in there that is not just peroxide, that's able to kill the microbes. And this means that this honey potentially has some really long-lasting medicinal potential, and it could even theoretically be heat sterilised for medicinal use. But wait, there's more. This honey also had an extraordinary amount of hydrogen peroxide. So what you can see here is a graph that shows the different honeys from the different species. Up the top, we've got time points. So we've got 1 hour, 12 hours, 24, going up all the way to a week. And the darker the colour of blue in this square indicates the amount of peroxide that was produced. So down here we have a Jarrah honey, which is a very high peroxide producing apis honey. And you can see that it produced about 50 parts per million, and that was done by 12 hours. Now, when we look at these stingless bee honeys, they just produce peroxide over an incredibly long period of time. So some of these were still going after an entire week, and they actually reached very high levels as well, like around 100 parts per million, which is about double what we were seeing in the honeybee honeys. So that's really amazing and unusual to see such strong and prolonged production of peroxide as well. So it looks like these honeys really have a 1-2 punch. They've got this peroxide production, and then they also have something else that's heat stable that is able to back that up as well. And then I went rooting around in our lab fridge, and I managed to find, amazingly, some T. carbonaria honeys that had actually been stored for 18 years. So these had been tested 18 years ago in 2008, and then I retested them in 2024. So on the left in these light orange columns, you can see the original tests. On the right, you can see our updated tests. And we found that all of that peroxide activity was gone. That's not surprising because 18 years is a really long time. The enzyme had degraded. But that heat-stable non-peroxide activity was basically exactly the same as it had been 18 years ago, which is quite amazing, really. We also took some of this honey and we spread it out on agar plates. So an agar plate's just like a yummy, nutritious surface for microbes to grow on. So we would expect that if there were any microbes in the honey, it would show up on this plate. This is an example of what kind of a typical honey plate would look like. This is one of our T. Hawkingsi samples. And this is what all of our 18-year-old Carbonaria samples look like. So barely anything growing on them at all, which is really amazing because they actually managed to remain sterile for that entire time. So what makes it special? Well, we did a lot of chemical analysis to try and figure it out. And here's what we do know. It is not methyl glyoxal, so that's the special component in Manuka honey. We tested for this and it was present at very low levels or not at all in our honey samples. What we instead saw was a very high concentration of phenolic and antioxidant compounds. So these are things that are coming from plants. It was about 2 to 3 times the level that you would see in honeybee honey. And we also saw some very diverse proteins and enzymes. So here on this graph, you can see these are our Carbonaria, our Hawkingsi, and our Austroplebia honeys. And this is a Jarrah honey for comparison. And this is the number of unique proteins that we detected in the sample. So basically, when we look at the tetragoniolar samples, we can see that on average, they had way, way more types of proteins than we found in our honeybee honey. So there's potentially something unique there that the bees are actually adding themselves. And of course, it also has a very low pH, so about 3.6 to 4.1, which makes it hard for microbes to grow. But this is probably not the main mechanism of action. It's just something that supports the overall action. So we think it's probably a unique combination of the bee biology. They are processing their resources in a unique way, or they're doing something unique, as well as the unique compounds that are coming from Australian plant sources that are responsible for this activity. And species differences were also important. So all of the honeys were great, but there were some differences between the species. So the Carbonaria honey had the highest hydrogen peroxide production, and it had the strongest heat-stable activity. The Hockingside honey had the highest phenolic content, so those were the darkest honeys. And in those honeys in particular, we saw a very strong correlation between the darker the honey, the stronger its activity. And the Austroplebia honeys, they had good total activity, but they were much less heat stable. So they weren't so good after we heated them, but they were pretty good fresh. And that potentially just says that, theoretically, different honeys may have better properties for certain types of medicinal uses. So what are those potential medical applications? Well, the most obvious one would probably be wound dressing. So the fact that these honeys produce peroxide over such an extended period of time is really beneficial for this. It means you'd have to change the dressing less often. And it's also got that backup heat-stable activity as well. So potentially under less than ideal conditions, if the wound isn't sort of, being treated very well, it might also have some backup activity, even if the enzyme that's creating peroxide is degraded. Another one would be skin infection treatments. We saw very good activity against those fungi that cause tinea, so it'd be really good as a topical treatment for that. The ultimate goal would be fighting drug-resistant bacteria, whether that's using the honey itself or using the honey to identify specific compounds that we can then go to synthesize independently of the honey to try and fight some of those drug-resistant bacteria. And the fact that it had such high levels of antioxidant compounds means that it could have some potential anti-inflammatory applications as well. And then, of course, it can withstand heat sterilization, which would be great for medicinal uses, and it seems to have quite a long shelf life, so it could potentially be stockpiled in places like rural and remote communities, which would be great for medicinal applications as well. Of course, you're probably all thinking, that sounds great, but can we actually make enough honey to really do anything with it? And you'd be 100% right, that is the main challenge. It would be scaling production. These bees are obviously quite small, and they make a very small amount of honey, so it's something like half a litre per year compared to, dozens of litres for honeybees. But that said, I think interest in these bees is growing. More and more people are keeping them. There was a recent Food Standards Australia, New Zealand legislation that came out that meant that this can now be commercialised and sold nationally and internationally. But what we would really need to do is more beekeepers, keeping more hives, and more knowledge about how to manage those hives to actually ensure that we're getting optimal medicinal quality honey. So what does this actually mean for beekeepers? Well, if you make native bee honey, this is just more scientific backing to the unique properties of this particular honey. It's never going to be a supermarket honey, of course, but it could be a very small batch but high-value premium product that has some scientifically backed properties behind it. And I wanted to say that this research generated quite significant media coverage. I say that not to toot my own horn, but to say that I think this shows that there is real public appetite for Australian honey stories. It was a very feel-good story. It's like Australian innovation, it's like Indigenous knowledge being backed up by modern science, it's like, natural products as a solution to modern problems. And I think even if you're just a honeybee keeper and you're not interested in native bees, I think that stories like this really raise the profile of the entire Australian bee industry. It gets people interested in bees, gets people interested about the amazing work that beekeepers do, and I think that it just positions Australian honey in general as a premium product. So I think a rising tide lifts all boats. Future directions. So theoretically, in the future, this is where this research could lead. This is, of course, contingent on time and funding and partnerships with people in the medical community, but this is what the future could look like. The key thing to do next would be to try and identify what those specific molecules are that are responsible for this unique activity. That would really open the door to what we call establishing the mechanism of action, or really characterising how this honey is killing bacteria and fungi. That could then go on to actually be used in some small-scale clinical trials to see if it is good for wound healing. When we identify those compounds, that means that we can then also standardize a commercial testing protocol, so we know what we're looking for, we can measure the properties of the honey better, and then we can go on to understand things like bee foraging patterns and bee health and productivity and how that actually influences the properties of the honey as well. And then all of that would ultimately lead to developing best practices for production so that we can ensure that we are producing sustainable and high quality medicinal honey. Okay, so often when I give these talks, someone will ask me the question like, that's great, but how do we know about this sooner, or how can we know when new things come out? And I guess the truth is a lot of this research just goes into scientific journals and it doesn't necessarily reach the people who would be most interested in it, which is beekeepers. So I've just started a small mailing list. If you're interested, you can scan that QR code and join. You probably get maybe 3 emails a year just when we've got a new paper or a new update that I think is worth sharing with you, both about native bee honey, also about honeybee honey as well. And yeah, with that, I'd just like to thank everybody who was involved in this study, particularly the beekeepers who provided samples and our funders who made this work possible. And thank you for listening.